Welcome to this broadcast of GFoRSS TV dedicated to food contaminant detection. Today's broadcast will be covering several facets of food contaminant determination as part of regulatory measures. Food contaminants continue to be an area of concern for consumers and for food business operators. And this is particularly the case for chemical contaminants. It is, in fact, those contaminants that are generally responsible for chronic effects, so generally effects that we are not able to see or that we are not able to identify as a result of an immediate health impact, but rather as a result of a longer-term exposure. During this broadcast, we will be focusing on identifying the regulatory environment related to the determination of contaminants in food. Why do we test what we test? Why do we have the requirements that we need to follow in order to have results on the basis of which decisions would be made? We will be also investigating some aspects related to the sampling of products that would be designated for testing in order to identify the level of some target contaminants. We will be also discussing the fit for purpose for methodologies that need to be selected in order to identify either one contaminant or a series of contaminants in food products. And finally, we will have the opportunity also to discuss some of the methodologies and the use of new technologies associated with the detection of low levels of contaminant, either for single compounds or for multiple compounds that may find themselves in food matrices. As you know, this area is an active area of research. There are a number of challenges and priorities, and the analytical community that is responsible for the investigation of food products organized itself in the form of networks. And one of the most important and useful networks in this regard are the networks that are set by AOAC International. We will have the privilege to have with us two leaders of AOAC International, responsible for the creation and for the management of the India section of AOAC International, Dr. Srinivas Joshi and Dr. Kaushik Banraji, who have taken the time to share with us their perspectives on challenges, priorities of contaminant testing, but also on what a community of AOAC International contributed to in fostering best practices and in addressing also the challenges of food contaminant testing. As was indicated in the first broadcast, the material that will be used to help us in navigating this topic is taken from a number of initiatives and interventions. You will note that some of the material is coming from courses that were developed as part of e-learning programs. And in this particular instance, I would like to highlight the program that was developed by the Global Food Regulatory Science Society in partnership with Laval University and the Platform of Food Risk Analysis and Regulatory Excellence. And that was developed as a result of the support and the partnership of the US Department of Agriculture, Foreign Agriculture Service, and the United States International Development Agency. This program was, in fact, developed as part of a capacity building initiative targeting a number of experts in the African continent, and we will have the opportunity to review some of the elements that were developed as part of this training program. We will have also some material that was extracted from previous webinars and programs that were delivered by GFoRSS in previous events and in previous webinars. Contaminant testing in food products is generally the result of compliance verification activities and conformity assessment programs. In most instances, we test for contaminants in order to respond to regulatory measures, either regulatory measures resulting from setting a maximum limit or a maximum residue limit when it comes to pesticide or veterinary drugs, or regulatory measures that are associated with regular surveillance and monitoring, attempting to determine the baseline levels of contaminants in food, on the basis of which we would identify potential deviations that would require attention and intervention, mostly in the form of preventive controls. So we will be reviewing the regulatory environment related to food contaminant detection and the importance of this aspect of analytical detection of contaminants in order to support us in decision-making processes that are based on risk analysis. This section will focus on the regulatory management of contaminants in food. The learning objectives are to acquire knowledge about risk-based regulations. Food regulations may be both hazard and risk-based. In hazard-based approaches, the presence of a potentially harmful agent at a detectable level in food is used as a basis for the risk management actions. Generally, this is a zero-tolerance approach, and a regulatory measure would be based on that. Risk-based approaches, on the other hand, allow consideration of exposure in assessing whether there may be an unacceptable risk to health, and this would be the basis of the risk management action to be taken. As presented in a previous presentation, food contaminants may cause immediate effects or may be associated with potential long-term effects after chronic exposure to the chemical. One example of an immediate effect is gastrointestinal illness, such as nausea, which can be caused by high levels of industrial chemicals. Examples of long-term effects include impaired cognitive development in children, chronically exposed to relatively low levels of lead, and liver cancer that may be resulting from chronic exposure to mycotoxins such as aflatoxin. However, the impact of food contaminants can easily be extrapolated to broader issues such as food safety, food security, and food waste. For all these reasons, there is a need for a regulatory framework allowing the products and the production of safe food to be set in a manner that allows the mitigation of the presence of contaminants in these foods. The primary purpose of food legislations and regulations are to protect the health of consumers. To protect the consumers from fraud or misrepresentation of the food, and also to ensure the essential quality and wholesomeness of foods. Food laws and associated regulations must first provide the legal authority and an adequate legal framework for the food control activities to be carried out. Generally, food law and the associated regulations would prohibit importation,
distribution of food products that are adulterated or have labels that are false or misleading in any context. This, in fact, applies to foods that are produced domestically and distributed, or imported from other countries and distributed on the local market. Food regulations can also be thought of as catalysts to developing countries' participation in trade. Regulatory measures that address risks in food production and food consumption would underpin the structure of market transactions within the country and would influence the competitive advantage among the various trade partners. The adequate implementation of the regulations would encourage fair trade practices through compliance with the provisions of the food law and the regulations. In fact, this would help protect the honest manufacturers, the honest dealers against unfair competition. It also, and when we say it refers to food laws and food regulations, help stimulate the development of the food industry because quality control based on sound scientific guidance tends to promote better consumer acceptance of foods and would allow for more market access opportunities. In other words, regulations are important for all stakeholders, including consumers, but also food business operators. And this helps all stakeholders to maintain trust in the capacity to produce and access safe food products. An effective food safety system requires effective regulations, policies, and procedures along the entire farm to fork continuum. It also requires the cooperation by the multitude of partners, including competent authorities, meaning government, industry at all levels of production, farmers, producers, processors, importers, exporters, and retailers, but also individual consumers. To illustrate this, governments or food competent authorities are responsible for establishing food safety requirements for industry to meet. These competent authorities would ensure also the compliance with food safety requirements, with these food safety requirements that are set potentially through regulations. The food industry is responsible for ensuring that food produced is safe and meets all the requirements established by the competent authorities, meaning government. Consumers have also their responsibilities, and they are in fact responsible for choosing foods that fit with their health condition, but also to handle these foods in a manner that would not increase the level of potential risks. Food safety regulatory requirements are intended primarily to reduce the risks of consuming unsafe food. The same objective is pursued by the international standard setting body that is the Codex Alimentarius Commission. The risk analysis process comprises three separate elements of risk assessment, risk management, and risk communication. It is widely recognized as the fundamental decision-making process, meaning the methodology that underlies a sound regulatory framework. A sound regulatory framework that is predictable and that allows the protection of consumers, but also the availability of fair practices in the food trade because of the values of predictability, of transparency, and systematic approaches that are followed. This is how the development of food safety standards is carried out. It is based, or it should be based, on the risk analysis process. According to the Codex Alimentarius Commission, risk analysis should be used to identify hazards, to assess and characterize their effects, the effects of these hazards, to establish the appropriate control measures, to prevent, of course, these risks, and also to communicate those risks and those measures to all stakeholders. Risk communication, in fact, must be included to present the analysis to consumers and to all stakeholders. At the global level, the risk analysis principles ought to be applied everywhere in the development of risk management measures for contamination in food. This is, of course, to ensure that food is safe and can be traded amongst countries based on the same foundation of the development of risk management measures. To start, it's very important that we make the difference between hazard and risk. A hazard is an agent in food with the potential to cause harm. On the other hand, a risk is the estimated probability and severity of adverse health effects in exposed populations that would be consequential to the hazard's presence in food. From a global standpoint, a food safety hazard refers to any agent with the potential to cause adverse health consequences for consumers. Food safety hazards occur when food is exposed to hazardous agents, which is the result of the contamination of foodstuff. Food hazards may be biological, meaning microbiological, for example, chemical, or physical. Sometimes some other categories are added, like nutritional and biotechnology-related hazards. It can also be considered as the perception of the potential impact of these hazards would make them as such. And this is particularly the case for biotechnology-related issues. In some cases, food allergens are separated from chemical hazards and are categorized as allergic hazards. So, each individual food contaminant is not a risk, but is identified as a hazard. And being able to detect and to identify the hazard is the first step in the risk analysis process. And this is where analytical methods and analytical processes are so important. They are important to help identify, detect, and quantify the potential hazard in food. The presence of chemical contaminants or other undesirable substances in food and feed is often unavoidable, as these substances may occur in a ubiquitous manner, like dioxins and dioxin-like compounds or heavy metals, or maybe also of natural origin, for example, in inherent plant constituents such as alkaloids or mycotoxins. That may be the result of the metabolic process of some fungi that are present on a crop, on a food product. This is what we saw in a previous discussion. Therefore, human exposure to such substances may be unavoidable. The risk assessment for chemical contaminants in food relies on the integration of two components: the knowledge about the human exposure to these substances via food and other routes, and their potential to cause adverse health effects. The risk characterization is the result of this analysis, and it is defined as the likelihood of the occurrence of the adverse health effects at a given exposure. To perform a robust risk assessment, there is a need for sufficient knowledge on human exposure, meaning the occurrence data of those contaminants in food, but also the level of consumption of these foods by humans. It is also contingent on being able to rely on a sufficiently sound toxicological database. Manufacturers do not provide necessarily toxicological information for most of the chemical contaminants that may be present in the foods that they make available, with the exception of substances that are intentionally used in conjunction with food production and for which there is an obligation to conduct a safety assessment and therefore to supply the toxicological database associated with these substances. This is the case for additives. It is also the case for substances that are indirectly added to food, like pesticides or veterinary drug substances. Chemical risk assessment is a well-established process, and in general, it would enable the assessment of risks from long-term and chronic exposure to a chemical. It includes the assessment of food additives, of residues of pesticides and other agricultural chemicals, residues from veterinary drugs, chemical contaminants from any source, and natural toxins such as mycotoxins and phycotoxins. When sufficient data are not available to support a quantitative assessment of the risks that may be associated with the food contaminant, a qualitative approach to characterize this risk may be the only alternative. The risk assessment process can include a variety of models to reach the conclusions. For example, the concept of acceptable daily intake, which is essentially a concept to characterize the hazard, may be considered as a component of the hazard characterization and therefore of the risk assessment. The hazard characterization process requires the knowledge about a specific hazard, including the identification of the toxic substance. It can be done for a group of molecules like dioxins or aflatoxins as a whole, for example. It also requires the knowledge about the metabolism by humans and animals as may be appropriate. Sometimes the culprit molecule becomes toxic only after certain modifications, as after, for example, a process of metabolization. Toxicokinetics and toxicodynamics are also relevant, like the information on the possible carryover of the toxic substance from feed to edible animal tissues or products. This is in fact the case for animals that may consume in their feed aflatoxins and which essentially may be vectors to carry over the aflatoxin in their tissues or in their fluids. So essentially, for example, in milk or in muscle. Another example may be food allergens. It is known that allergic consumers will not react the same way to the exact same dose of allergens. So some of them will react when they ingest a few milligrams of food allergens, while others will only react after the ingestion of several grams. So you have differences in the dose that may induce the response. You may also have a difference in the speed of the development of the response. All these parameters are to be accounted for in the development of the hazard characterization. Obviously, information about acute and long-term toxicity and other relevant toxicity data are important and should be considered. Finally, integrated toxicological expert advice regarding the acceptability and the safety of certain intake levels of contaminants, including information on any population or groups that may be specifically vulnerable, for example, infants, for example, the elderly or the immune compromised, should also be included in the data that will help to characterize the hazard. When there is an uncertainty, this uncertainty should be documented such that it is accounted for in the overall process. The other important step of the risk assessment is the exposure assessment. The exposure assessment requires the reliance on consumption data. These data are to be generated by using tools like surveys and should include the different populations of a country or a region. For example, if you take a country in, let's say, North Africa, you would need to have consumption data from people close to the Mediterranean Sea, but also from people who live in the Sahara. In the same way, in countries with mountains, isolated populations should not be excluded. On the contrary, these populations may often have a different risk profile because they often have a specific type of diet that needs to be accounted for. Evaluating exposure requires occurrence data as well. Occurrence data is the occurrence of the hazard in the food, which can be obtained through what we know as total diet studies. This is in fact a food surveillance program that monitors the concentration of chemical contaminants in food that are typically consumed by a given population. Foods are to be prepared for consumption as if this preparation would happen at home. Basically, foods are analyzed in the way they are consumed. This provides realistic information about the level of contaminants that may be present in the condition of the exposure from the diet. Preparation steps include washing, peeling, and cooking the food product, and several types of foods are combined into composite samples in order to analyze the given hazard in those food products. Exposure assessment is in fact the result of a product, the product of the occurrence data multiplied by the consumption data. So essentially, exposure equals occurrence times consumption. This leads us to estimate what we call the dietary intake of a given contaminant from a food source or from all food sources. Once the hazard characterization was conducted and the exposure assessment is estimated, the next step is the risk characterization. It is defined as an estimation of the probability of occurrence and severity of known or potential adverse health effects in a population based on the steps that we mentioned earlier, meaning the hazard identification, the hazard characterization, and the exposure assessment, leading to this risk characterization. Once the risk assessment is performed, the risk now must be controlled or mitigated via what we call risk management. Risk management is defined by the Codex Alimentarius Commission as the process of weighing policy alternatives in light of the results of the risk assessment and, if required, selecting and implementing appropriate control options, including regulatory measures. The goal of the risk management process is, of course, to establish the significance of the estimated risk and to compare the costs of reducing these risks to the benefits gained, and to compare also the estimated risks to the societal benefits derived from incurring the risk. It is also to carry out the assessment of the societal, political, and institutional impacts of the measures that may be considered to reduce this risk. The control of chemical contamination that is potentially present in food continues to evolve. But there is a key part of the current process that is always subject of attention, which is the international harmonization of the controls of contaminants in food. And to this end, the Codex Committee on Contaminants in Food, or CCCF, developed a Codex General Standard for contaminants and toxins in food and feed. Risk management strategies vary depending on the situation. These type of actions may include providing advice and guidance to consumers on the risks and the benefits of particular food choices. For example, consumers should be made aware that choosing non-predatory fish would help reduce the risk of exposure to mercury. Those risk mitigation measures may also include providing direction on how to reduce contaminants level. This is the case, for example, for food processing induced chemicals, where codes of practice may be developed in order to reduce the conditions of the occurrence of that food processing induced chemical. A classic in risk mitigation strategies that is used for contaminants in food is, of course, setting maximum levels for these contaminants. This is the case for several mycotoxins, for pesticide residues, where we set maximum residue levels now, but many other food contaminants. For certain substances, we may use what we call a zero tolerance approach. This means that no amount of the substance in question would be considered acceptable in food. This is generally the case for adulterants that may be intentionally added and that we need to keep away, to keep out of the food supply chain. This is the approach that we call a hazard-based approach. A hazard-based approach may materialize in the form of a zero tolerance approach. Now, implementation of the management decision should be always followed by monitoring of both the effectiveness of the measure that is chosen to control the risk, but also of its impact on the risk to the exposed consumer or to the exposed population. This is, of course, to make sure that the food safety objective that was pursued originally by the entire process is in fact being met. Among the several options that risk management offers, the development of maximum levels for chemical contaminants in retail food is an important one. Maximum levels are established by food regulatory agencies and should be enforceable by the same agency or another agency, which means that there is no interest to establish a maximum level which cannot be achievable by producers, or that could not be detected by currently available technologies for analytical methods. However, and even if a safety concern is identified and management action is required, the establishment of a maximum level may not necessarily be the best approach to pursue for the reduction of the risk that is associated with the foodborne chemical. For example, the presence of a contaminant in food may be the result of an incident that was temporally or geographically isolated and that could have been avoided. In this case, the appropriate risk management may involve the removal of the contaminated food from the retail shelves and taking corrective actions at the food manufacturer or the farm level to ensure that such contamination does not occur again. Short-term monitoring to ensure that the corrective action is successful would be required. In this case, in fact, it's very clear that there is no need to establish a maximum level in order to manage the risk. Maximum levels are in fact established in an effort to reduce exposure to a particular contaminant, and exposure is affected by the concentration of the chemical in food and the amount of the food that is consumed. Therefore, both the concentration and the amount of the food that is normally consumed must be considered when we develop a maximum level. As a result, in fact, a maximum level for a particular chemical may be different depending on the food for which it ought to apply. When we establish maximum levels for contaminants in food, the primary concern is, of course, human safety, although the availability of the food, the nutritional value, and the importance of the food in the national diet are also important considerations. We cannot establish maximum levels that wipe off the entire supply of a food product from the market, for example. There are, in fact, limited instances where we use or where we should use maximum levels as risk management measures for contaminants in food. We establish, in fact, an ML for situations where consistent adherence to the ML would help drive the overall human exposure down. So MLs should only be set for foods that contribute significantly to the exposure. Another reason where an ML may be useful is, in fact, in situations where we are trying to have a systematic approach for the industry to apply in order to manage the food, or for the regulator to check on the presence of the chemical. In fact, the most important approach that should be applied for the management of chemical contaminants in food is what we call the as low as reasonably achievable approach, ALARA. And this generally is by making sure that all measures are taken in order to prevent the points of introduction of a chemical contaminants in food, and also by relying on regular monitoring and the assessment of the risks to ensure that it is always maintained as low. The levels of contaminants in food should be monitored through regular surveillance activities by regulatory agencies, and surveillance data are used to help identify the potential contamination issues, and when warranted, we would conduct a risk assessment, and if the risk assessment conveys that there is a need to take action, then risk management strategies should be developed when that situation warrants that. So in fact, the absence of a maximum level for a particular chemical in food does not mean that there is no risk management approach or no protection. Monitoring and regular assessments for chemicals are one of the key measures to be adopted by food competent authorities to manage these contaminants in food. In fact, sometimes when you set an ml where you can reduce the levels of the contaminants even lower than the ml, setting a maximum level may have the detrimental impact to give a license to pollute to certain producers of food that would potentially use the ml as a license to increase the level of contamination, whereas there is a possibility to reduce those levels by the mitigation measures that are available to them. Now, risk management is not a project to complete or a task to check off on a to-do list. It is an ongoing process, and it should become part of a continuity of improvement culture. As with most activities, continued attention provides better and more efficient execution. Less efforts overall would be devoted to what we're trying to accomplish, and better results are generally achieved. Monitoring risk management strategy is actually one of the most important activities in the risk analysis process, which in fact is a cycle approach. Monitoring and review are the gathering and the analyzing of data so as to give an overview of compliance with food safety requirements. It includes the verification of compliance with the risk management decision by ensuring that stakeholders implement the options that were chosen. This also includes the determination of the ongoing effectiveness of risk management measures and the identification of any changes that would impact the risk analysis. Ongoing review of the risk mitigation plan is required to ensure that it is meeting the needs of the regulatory framework for which it's set. This review concerns all mitigation strategies, including the status and the effectiveness of the actions that were taken. Ensuring that all requirements of the risk management plan are being implemented is critical. Otherwise, the mitigation strategy can become an unconscious acceptance of the risk and may be identified as an additional risk itself. Now, risk communication is an important and a final element of the risk analysis process. The Codex Alimentarius Commission's definition of risk communication is that it is the interactive process of exchange of information and opinion on risk among risk assessors, risk managers, and other interested parties. A definition with a broader scope is the one that was adopted by the United States National Academy of Science, and it defines risk communication as the interactive process of exchange of information and opinion amongst individuals, groups, and institutions, which involves multiple messages about the nature of the risk and other messages not strictly about risk that express concerns, opinions, or reactions to risk messages or to legal and institutional arrangements for risk management. So in fact, it's encompassing all aspects of the risk analysis framework. Communicating the results of risk assessment and risk management serves many purposes. The quality and safety of food depends on responsible action by all involved at all stages in the food chain, including consumers. Consumers, in fact, require access to adequate information about potential hazards and appropriate precautions to be taken in the final preparation and serving of food. In addition, consumers need to be aware of and to understand the food safety control measures that are implemented by the competent authorities that oversee the food in the interest of protecting and promoting their health. Finally, all the regulatory frameworks in the world need to be updated from time to time, and this is also the case for risk-based frameworks. The monitoring of contaminants in food and the continued surveillance of foodborne diseases should help us identify potential new problems as they emerge. A risk analysis strategy will not be effective unless there is a mechanism that allows the identification of these new risks, and that it allows us, in fact, to track these risks through information and communication collected from stakeholders and from all sources of information. Being able to update the list of emerging hazards is a critical part of maintaining an effective risk analysis strategy. Now, the modus operandi of risk analysis is always evolving as new scientific data becomes available in order to help us improve the hazard characterization, the exposure assessment, and the overall risk assessment. Basically, there is always new data that we need to be aware of that would help us enhance our better understanding of the risks. It is essential, therefore, to adjust our approaches based on these new data and to help us, for example, reprioritize the risks accordingly. It's also always a good idea to put the previous assumptions to the question and try to validate these assumptions every time that there is new information that is available. There may be also the need to develop new assumptions as a result of new information that becomes available. In the same way, new mitigation options could be developed when a new risk characterization is developed or is produced. When there is a change on the risk or the probability, it may make sense to adjust the risk management approach that is already in place. This is again to reiterate the fact that the risk analysis framework promotes an evolutionary process in order to keep up with the risk and to ensure that consumers and consumers' health in particular is continuously protected and protected in an optimum fashion. This brings us to the end of this part of the course, where we have reviewed the importance of the risk analysis framework as a foundation to analyze, but also to manage the risks associated with contaminants in food. We have identified that the ability to detect these contaminants is a crucial part of the risk analysis process, not only in the early stages, so the first steps of the identification of the hazards, but also in the assessment of the effectiveness of the risk management approaches that were adopted in order to mitigate these risks.
When we are faced with the need to determine a contaminant in a food product, one of the key considerations that we have to ask ourselves about is what is the method that is the most appropriate and the most adequate for the objective that we are pursuing, and particularly for the regulatory objective that we are pursuing. Are we looking to analyze multiple samples in a very short time? Are we looking to determine the result with accuracy and precision? Or are we actually looking to determine a semi-quantitative or qualitative result? All these considerations may dictate the approach that we would have to follow to select the analytical methodology that would be most suitable to achieve the objective pursued. This will be the focus of the next segment.
This section will review the various parameters that we need to consider to choose an adequate method of analysis. From a regulatory perspective, food products have to be tested to verify their compliance with regulatory requirements or as part of monitoring programs. Similar requirements may exist as a result of private certification needs, which may mandate similar level of assurances and therefore the associated verification. Food analysis is an important cornerstone of risk analysis, as we discussed earlier. Risk assessment cannot be conducted unless it relies on data obtained as a result of the analysis of food products. The reasons to analyze food products may be diverse, from quality control to supporting research and development. The need for robust and accurate analytical methods is ever more important, but what is also crucial is to be able to select a method that fits with the purpose of the testing. Numerous methods are often available to test for food samples for a specific food contaminant. There is a need to differentiate what we call a method from a technique. By definition, a method is the application of a technique for a specific analyte in a specific food matrix. So, you can see various equipments from liquid chromatography to gas chromatography, mass spectrometry that can be coupled with either LC or GC, which are both techniques. But these techniques can be used for the development of a number of methodologies for the quantification of pesticides in barley, for example. That would be a method. A procedure that explains how to apply a method to a particular sample, including information on obtaining the sample, handling the interference, and validating the results. That procedure is needed in order to reach the quality requirements of the method. In the same way, a protocol, which is a set of guidelines specifying a procedure that must be followed, have to be created if an agency is to accept the results. Protocols are common when the results of an analysis supports or defines public policy. So, for example, a regulatory requirement. Using a fit-for-purpose approach relies on the assessment of method performance to define the suitability of a given methodology to provide an answer to a given question or a given requirement. To select methods used to determine or quantify food contaminants in some food samples, one must use analytical methods that fit their intended purpose. A fit-for-purpose approach includes several components, several questions, like the analysis of the compounds, of course, but also the sampling, the sample preparation, and the handling or the treatment of the results. In order to analyze a sample, the first question is always, does an analytical method exist, or do we need to develop it? As explained in previous discussions, there is no point to regulate a food contaminant or to mandate a maximum level, for example, if we're not able to detect it with an analytical method. Consequently, when there is a regulatory requirement for a specific contaminant in food, there should be an associated method that has been developed and validated to detect and quantify the contaminant in the food in question with the achievement of the level of detection that is required. Now, on the other hand, if the purpose of the analysis is to monitor the contaminant in food and support the application of what we called the as low as reasonably achievable principle, the ALARA principle, in this case, the method of analysis that should be selected ought to be as sensitive as possibly or technically achievable. That is why we note that methods that are generally used in total diet studies tend to be far more sensitive and hyphenated than the methods that are applied in the context of compliance verification programs, for example. Several non-for-profit scientific organizations have compiled and published methods of analysis for food products which have been carefully developed and standardized. The AOAC International, so the Association of Official Analytical Cooperation at the international level, provides guidance on methods for almost most of the food contaminants that we discussed previously. And this may be applicable for a very large range of food products. What we call official methods are generally developed and are intended for use by industries, for their compliance verifications. They are also intended for use by contract laboratories. They can be also applied by a number of regulatory agencies. AOAC International has also programs to develop standards, to test the performance of commercial tests or commercial kits, but also to check the laboratory proficiency. Now, AOAC has a number of other partners that operate in the same field of standardization. ISO, so the International Standardization Organization, but also some national organizations like the Agence Française de Normalisation, AFNOR, have also published a number of methods of analysis. In addition, there are several organizations that, for example, specialize in certain foodstuff, and they provide methods of analysis for the crops or the commodities that they tend to represent. Now, when you have several options of methods of analysis that are available, you need to make a choice that fits your purpose and is adapted to your needs. And it is important that the data that is generated from the food monitoring, for example, yields acceptable trueness and precision for use, particularly when it is used in complex risk assessment calculations and statistical analysis. In the same way, regulators who enforce maximum residue limits, MRLs, must have the confidence that the reported analytical results represent the true residue concentrations. Equally important in quality assurance programs is the reliance upon a well-designed sampling plan, in the absence of which results may be considered as unrepresentative and therefore invalid for use. Now, let's discuss the fit-for-purpose approach. The requirement of the analysis will determine the best method, and to choose a method, preference should be given, of course, to methods of analysis for which the reliability has been established in respect to several criteria. Now, the criteria that are shown here, accuracy, sensitivity, selectivity, robustness, ruggedness, precision, intra-laboratory repeatability, inter-laboratory reproducibility, and many more, are part of the criteria that have been described extensively in the scientific literature and represent criteria of choice. All analytical methods provide some variability in the results, and it is important that the methodology that is chosen is done such that this variability does not impinge on whether it can answer the question that is the key objective of the measurement. For example, if a methodology is very susceptible to minor changes in temperature or timing, it will probably not be practical to use this method in a production or a field testing environment, because it will be sensitive to that, and therefore we will compromise the accuracy of the results. However, this same methodology that is sensitive to temperature and so on, could be perfectly acceptable if it were to be used in a temperature-controlled laboratory environment with highly trained personnel, highly trained technicians, for example. All analytical measurements have a range of analytes that they are accurate for, and it is important to choose a method that ensures that the value of the analyte of interest falls within the range that the method applies for. Measuring analytes outside of this range can lead to erroneous results. So, for example, if the analyte is present below the limit of detection, or on the contrary, if it is, for example, so high that it would be outside of the calibration curve, it will give inaccurate or variable results. Importantly, it's not always necessary to chase zero. If all that is important is the knowledge that a contaminant is present below a certain value or not present above a certain level, then it's not always necessary to quantify the analyte below a given target. That's why we don't have always the need to apply the most sensitive analytical method, or what we call the best analytical method, but rather use one that achieves the sought after goal. The following questions are generally asked and have to be answered before selecting a method and actually starting the analysis. Generally, the question is Can the method be carried out in a timely manner? Does the cost of executing the method or using the equipment involved would be too onerous? And can the method be executed by trained staff in a relied fashion? Those are some of the key considerations that need to be looked at when selecting a method as well. Now, several considerations regarding the contaminant may dictate also the choice of a technique of analysis. Now, we're talking about the selection of the technique. So, for aflatoxins, for example, you may use lateral flow devices or an ELISA test, as well as an LC-MS technique, amongst others. For the detection of peanut allergens, the same thing. You may have the choice between a lateral flow device or an ELISA. LC-MS might be a technique that could be suitable, even though it's not so practical for quantification purposes. For heavy metals like arsenic, generally the techniques that are available are quite limited, and ICP-MS may be the most used technique so far and probably the best fit for this kind of contaminant. When we're looking at pesticides, and if we are looking at, for example, a single pesticide like DDT, we may have options like liquid chromatography or gas chromatography coupled with mass spectrometry. But again, when we are looking at a singular pesticide, the reliance upon ELISA techniques may be a suitable option to consider. Another very important question is, what is it that you're trying to look for? What is the analyte, or what are the analytes that you're trying to detect? Now, it may look like a silly or a very trivial question, but in fact, it is a key point. You have to remember that regulatory requirements may be different from a food product to another. For example, the series of mycotoxins that may be found and that need to be checked for wheat or barley may be different from mycotoxins that are found in other cereals. Consequently, the choice of the analytical method would be different. When you have to detect mycotoxins, does it mean all mycotoxins? Does it mean a subgroup of mycotoxins like aflatoxins? Or are you looking at a specific mycotoxin, so for example aflatoxin B, in this particular subgroup? The answer to this question will impact your choice of the methodology and also the choice of the technique. If you need to detect a single mycotoxin, a technique like ELISA might be suitable and might be enough. But if you are looking to detect multi-mycotoxins in a food matrix, a better fit would be the selection of a technique based on liquid chromatography and mass spectrometry. Another key point is that the selected method should be chosen such that it is the least affected possible by other components in a sample. This is an important consideration because it really ensures that the result is an accurate reflection of the question that is being asked. For example, there are potential inhibitors in food matrices regarding ELISA tests. So they inhibit the performance of the ELISA, the response of the ELISA, leading to false positives or false negative results. In the same way, some interference can lead to inaccurate results even in LC or GC-based methods. Another extremely important point is the sample preparation that could be completely different from one food matrix to another. A good example is methods for pesticide detections, which provide sample preparation for the various types of foods based on their composition in water and lipidic content. As you may imagine, the detection of pesticides in hops or meat would need different extraction protocols. In the same way, water samples may only need a very light extraction process. Now, methods for the sample preparation of food should accomplish a number of the following functions, at least one of them, if not more. The ability to separate some of the target molecules from the food. So essentially, try to get away as much of the food ingredients as possible in order to have the target analyte in, I would say, as clean a fraction as possibly be. It may also want to accomplish the increase of the concentration try to purify from the extraneous material, achieve a volume reduction, produce a homogeneous sample, and exclude as much as possible of the inhibitory substances. Now, finally, another key step that is important to remember in food analysis, and that is the sampling procedure, which is an important consideration that greatly contributes to the reliability of the results and the final decision of compliance or non-compliance, for example, for an entire food batch. To address the problems that are associated with sampling, many sampling plans have been developed and employed based on statistical parameters that balance consumer safety and the protection of producers. With the exception of liquid food samples, such as milk, for example, or highly processed food, traditional sampling plans or methods for foodstuffs are usually not suitable for several molecules, like mycotoxins, which may be present in a not so homogeneous manner in food. In fact, mycotoxins are likely to be present in some pockets of a bulk food, for example, and therefore you have a very non-homogeneous product. Due to this uneven distribution of the analytes, like mycotoxins, it's very challenging to get a representative sample from a bulk food. That's why it's very important to consider a sampling plan that needs to be implemented that would allow to check samples that would be representative of the entire bulk food and that would therefore guarantee the trueness of the results. It's important that data generated from food monitoring yield acceptable trueness and precision for use in complex risk assessment calculations and statistical analysis. In the same way, regulators who enforce maximum residue levels and maximum limits, for example, must have the confidence they need such that the reported analytical result represents the true residue concentrations or the true value of the contaminant. In quality assurance and research activities, a precise estimate of the true contaminant concentration in a lot becomes important, and the absence of a well-designed sampling plan will result in the collection of unrepresentative samples, which may invalidate the results of subsequent contaminant determination. Now, a sampling plan is defined by a test procedure and a defined accept-reject limit. A test procedure is a multi-stage process and generally consists of 3 steps: sampling, sample preparation, and quantification, or what we know generally as the analysis, commonly used the word analysis. The sampling step specifies how the sample will be selected or taken from the primary sample to the analytical sample. Regulatory agencies, including guidance from the Codex Alimentaris Commission, developed a number of guidelines to perform this sampling for the determination of a number of contaminants in food and feed. This is particularly relevant when you have that uneven distribution of the contaminants in a bulk food. In fact, what you see on the slide comes from a Codex guidelines and represent a generic sampling plan. At the end, the measured contaminants concentration in a sample are used to estimate the contaminants concentration in the overall bulk lot, or compared to a defined accept-reject limit that is usually equal to what we consider to be a regulatory limit. Because of the uncertainty that may be associated with the sampling plan, the true food contaminant concentration of a bulk lot may not be determined with the full certainty. Nor can all the lots, in fact, be correctly classified into good or bad categories with the level of accuracy that may be needed. This concludes to the end of the current section of our learning plan. I hope that through the discussions that happened earlier, we were able to review the various classes of food contaminants, how they are managed, how they can be regulated, and the importance of analytical methods in order to apply these risk management approaches. The discussion of the fit for purpose is an important consideration to select the analytical method, the analytical technique, and equally important are the aspects that surround that methodology, including the sampling plans and the sampling preparation.
Fostering collaboration amongst members of the food analytical community, particularly in the area of chemical contaminant detection in food, is a key condition of success, particularly in a field where we witness priorities emerging on a regular basis and challenges being faced by the food analytical community. This is what AOAC International offers as one of the value added in terms of being part of a community, of a food analytical community. We will have with us two key leaders of AOAC International who contributed in the creation and the operation of AOAC India section. Dr. Kaushik Banerjee and Dr. Srinivas Shoji will be with us and will share their perspectives on the challenges, the priorities, and also the opportunities offered by AOAC International and the communities it fosters.
Well, we are fortunate to have with us two colleagues, two opinion leaders and eminent experts in food contaminant detection in Dr. Kaushik Banerjee and Dr. Srinivas Joshi. In fact, we have here main and major contributors to the AOAC community working in food contaminant analysis. Dr. Kaushik Banerjee is the chairman of AOAC India section and the former president of the same section. Srinivas Joshi is the current president of AOAC India section, and it's very rare to have essentially the current and the former leadership of the same section being interviewed at the same time. So thank you very much for taking the time to be with us today in this broadcast. And what we would like to ask you and perhaps have you share with our community of viewers is really your experience in food contaminant detection, the challenges, the priorities, and also your experience in shaping the food analytical community in India. And perhaps starting with you, Dr. Banerjee, basically, if you can share with us, based on your extensive experience in this area, what are the main challenges that you see in setting up capacity for food contaminant detection overall?
Thank you, Professor Samuel Godfrey, for this question. It's very interesting, and of course, we all know that there are several challenges, and challenges are almost common for most countries. Challenges are many. We have diverse food contaminants. There could be pesticides, mycotoxins, veterinary drugs, antibiotics, and so many emerging contaminants. The maximum residue limits are becoming very, very low. So access to appropriate equipment is becoming a challenge that which can really detect and quantify the residue levels as per the regulatory requirements, because if MRL is not available, then it is usually set at default level. Then we have a problem of non-availability of trained human resources, especially in developing world. We require a lot of training, capacity building to develop their knowledge about equipment handling and their knowledge about the latest guidelines of method of how the analytical method quality control should be maintained. Then we have emerging contaminants, which every year they appear, and we deal with them, and that's a major challenge that we have to set the new methods. We have to implement those methods in the testing labs. And of course, another important point which currently we are facing is the MRLs for processing processed products. And whenever you detect any pesticide in processed commodities, many a times processed commodities do not have appropriate analytical method that is set. And then if something is detected, we do not have the processing factors that are required for risk assessment and risk management purposes. So these are in brief what I felt that we require to develop capacity in these fields across the countries. Thank you.
Thank you for that, Dr. Banerjee. And I guess in some of the items that you mentioned, you've highlighted the evolutionary nature of this field of food contaminant testing, and the fact that there are priorities that are emerging, issues that we were not necessarily paying attention to, or at least giving that much of importance. So this brings me really to discuss priorities in food contaminant testing and the way this is being evolving over time. So Dr. Srinivas Joshi, if I can ask you to give your perspective on the evolutionary nature and also the priorities that you see right now in food contaminant testing, and perhaps even provide us with some foresight on where we should be looking at in the future.
Thank you, Dr. Samuel. And it's a great pleasure to have a discussion on this platform. When we see the food contaminants, typically the evolution, just to quote one evolution pertaining to the India, in 2004, we were monitoring 12 pesticides prior to 2004. And now currently we are in 2022, we are monitoring somewhere close to 310 pesticide residues and its metabolites put together. So you can see in last 20 years, there are more than 30 folds growth in the monitoring system. So it is very, very vital that testing plays a very vital role for enforcement of the regulations. Either it's a domestic, import, or exports. And over the years, what we have seen, the generic application contaminants and residues, what we say, the generic application areas are pesticide residues, veterinary drugs, and mycotoxins. That every lab nowadays has to develop a competency at least in these three application areas. And the futuristic, what we see is more of kind of a persistent organic pollutants in terms of compounds like dioxins and furans, PCBs, polychlorinated biphenyls. So these are the futuristic kind of approach. And over the years, whenever we have seen year after year, there are new list of compounds which are added. Either it's a part of a trade requirement, it's a part of the mandatory regulatory requirement. So laboratory has to build up the capacities to cope up with these requirements. That means they have to develop the methods, they have to validate the method, and generally they need to demonstrate the robustness of the method. And from India section of AOSC, we have taken an initiative pertaining to some of the challenging applications or the challenging matrix vis-a-vis the challenging target compounds. What we try to do over the years, in last few years, we have tried to pick up some of the challenges which are typically typically key requirements for the regulators. Are we try to develop a single lab validation for certain specific target compounds vis-a-vis the matrices, and try to verify it with a multi-lab validation. The whole purpose is whenever we put in, we try to develop a method, validate a method, the whole purpose is at least 70 to 80% of the laboratories should be able to adopt this method. So that's what is our whole purpose. And capacity building is a continuous process, because there is a lot of evolution, a lot of scale up in terms of the list of target compounds and understanding of those target compounds. And as the number of compounds keep on growing, there is always a need to have a multi-residue approach. Either it's in pesticides, veterinary drugs, or mycotoxins.
Well, thank you for sharing this perspective, Dr. Srinivas. And in fact,
By just listening, in fact, to you and to you both, we identify how challenging, in fact, this field can be. And when a field like this is so challenging, collaboration, cooperation, creating networks tend to be one of the responses to offer to address these challenges and to be able to help each other in a community, in an analytical community. And in fact, what better than AOAC and AOAC International as a community that was created since over a century now, over a century ago, and that is trying to bring together the community of scientists. Now, you both have been the proponents and key actors in the creation of probably, by many testaments, one of the most active geographic sections of AOAC International, and that is the India section of AOAC International. If I may ask both of you to give your perspectives about what the section is bringing to the food analytical community in India. What is the value added? And perhaps even what are the challenges in shaping a food analytical community like this in a country as diverse and as important in terms of food production as India is? So here I'll leave you both really the opportunity to comment.
Yeah. So what I would like to add here, that basically we are trying our best so that each and every lab in our geographic region, they perform with equivalent proficiency. So that's a very important thing, that there are hundreds of labs in Indian subcontinent, and we expect that each and every lab should perform with the equivalent accuracy and precision in their analysis, so that the results should speak about competence, and that helps in regulatory compliance. In addition to that, the India section is bringing all the labs in a single platform, so that they can discuss among themselves what are the challenges. We are helping them in participating in various interlaboratory comparison programs, in proficiency testing programs. We have supported our Food Safety Standards Authority of India to organize proficiency testing rounds across the food labs in the country in various aspects, which not only include contaminants, but also include various nutritional parameters. And then we are trying to help them in developing their capacity by organizing various training courses in collaboration with the technology providers. Every year, our annual meeting, we try to include and involve the young professionals who are students, who are doing PhD, they are postdocs, and the early career professionals who are in chemistry and various food safety disciplines. And we are helping them in developing their capacity. And I'm very happy to mention that in AOAC International last couple of years, including this year, we have found that the youngsters from India have got the highest level prizes in AOAC Internationals, like Rising Star Award and such kind of things, which clearly indicates that our people there, their capacity is getting evolved. And we are also supporting various other countries which are located in the nearby regions. So whenever they feel any trouble in improving their capacity, we are supporting them. So it's a sort of holistic approach, a comprehensive approach that we are taking in capacity building. And when capacity is built, I feel that everyone comes to an equivalent platform. And that probably we need to implement food safety across the globe. Thank you.
Thank you, and congratulations for all the work that is done. I don't know if you have any additions, Dr. Joshi. So to
like Dr. Kaushik mentioned, effectively, we believe that any food safety initiative, any food safety testing initiative, or a food quality testing initiative should have a public-private partnership approach. neither only governments cannot do, neither only food business operators cannot take initiative. It's a collaborative approach between the our approach is basically with the regulators, with the academia, with the research organizations, the food business operators, and the contract testing labs. So what we call it as it's a total stakeholders who work together for the betterment of harmonized analytical methods. And this is a kind of a approach which is helping our country, as well as the India section is seen as a key resource partner for them, either in terms of the new method development, either in terms of the capacity building, either in terms of the awareness program. So we have quite a lot of programs what we do. And in on this platform, I want to mention that in 2021, India section was named as the world's best section also. So we received a world's best section award. So we have for this, all the stakeholders who are involved in the process have put in a lot of efforts to try to make that this is the section which is sustainable, viable, and helpful for the whole of the analytical community.
And I would say that's a very well-deserved title, and definitely an example to be followed by various members of the food analytical community and the various sections of AOAC Internationals. Again, congratulations for all the work that you do. Very thankful to both of you for taking the time in your busy schedules to be with us in this broadcast. Dr. Kaushik Banerjee, Dr. Srinivas Joshi, the current leadership of AOAC India section, and also members of the Global Food Regulatory Science Society, both of you, and a founding member and a member of the board of Dr. Kaushik Banerjee. So again, thank you for being with us.
Thank you.
You're most welcome. Thank you very much.
Thank you.
The results of food contaminant detection is highly dependent on the way the food has been sampled. And for a number of contaminants, it matters very much how the sampling has been carried out, particularly when you have situations where the contaminant may constitute pockets of high presence in certain parts of the food or certain parts of that food that is available for conformity assessment or for compliance verification. In this next segment, we will review the importance of sampling in food analytical detection procedures. This segment is introduced by Dr. Kerry Mon from Trilogy Laboratories and is part of a previous broadcast that was made by the Global Food Regulatory Science Society in a webinar carried out in 2021.
Good morning. My name is Carrie Mowney, and I am from Trilogy Analytical Laboratory in Washington, Missouri in the U.S. And on behalf of Trilogy, I certainly appreciate the opportunity to share some mycotoxin information about mycotoxins in general, as well as some tips and techniques that will help build a solid testing program in your facilities. First, I would like to take a moment and give you a little background on Trilogy. Our company was formed in 1999, and since our inception, we have had a solid emphasis in mycotoxin testing services and also offering products that support toxin testing. In the U.S., we handle a high volume of mycotoxin analyses, and we run between 200,000 and 250,000 analyses annually. We have our ISO 17025 and our ISO 17034 accreditation, and our facility also holds an ISO 9001 certification. We do have several facilities around the world, and that has allowed us to broaden our range of services and activities and our products. In addition to the U.S., we have facilities in Latin America and Europe, and a 2021 expansion opening in Ghana. As we all know, mycotoxin testing is a critical part of global trade. And unfortunately, this gets more complicated because many countries have different mycotoxin regulations. Imports that don't meet in-country standards, as well as any exports that are not within the recipient's specifications, have a financial impact that can be a huge burden for both importers and exporters. Additionally, the imported commodities headed for both human and animal consumption must be taken into consideration health consequences that are associated with those mycotoxins. Mycotoxins are chemical compounds produced by molds. There's actually hundreds of mycotoxins. One such toxin that we all know about is penicillin. Sometimes at the lab we will get asked how to kill a mycotoxin. This is hard to think about sometimes because we consider that the mold can actually be killed, but the toxin, like any other compound, such as, say, salt or sugar, can't be killed. Mycotoxins have been around since molds began growing. In fact, one of the plagues was thought to have been a mycotoxin called ergot. So, where exactly do these toxins come from? Well, different weather conditions produce different molds, and each mold produces different mycotoxins. So weather conditions typically are associated with different toxins. For instance, the molds that produce deoxynivalenol, which is also known as DON, or vomitoxin, typically prefer wetter, cooler conditions. In addition, plants are more susceptible to mold invasion during specific times in their growth and development. Any broken kernel, damage from insects, immature kernels, etc., will allow entryway for molds. And once those molds are in, they have a food source and they can then produce toxins. This type of contamination happens in the field prior to harvest. However, improper storage and transportation can also increase molds and mycotoxin production dramatically. Mycotoxins therefore enter the food chain both pre-harvest and post-harvest. Managing both points of the process is essential for solid risk management. I mentioned there are hundreds of mycotoxins. However, only a few are what we would call common mycotoxins with which we should be concerned. Mycotoxins are associated with weather conditions, but they can also be associated with specific commodities. The chart below shows the toxin and commodity associations as well as the general weather conditions associated with each mycotoxin. You will notice that corn is associated with all of the toxins. Corn has a wide production area and it is grown in many climate conditions. Wheat, on the other hand, has a more limited toxin association. The most common toxins associated with wheat are DON, zearalenone, and ochratoxin. One thing to remember, the processed products associated with the main commodity may also contain mycotoxins. In fact, many processes concentrate the mycotoxins. For instance, in wheat, the wheat mids, which are also known as the middlings, are that outer portion of the wheat kernel typically have the highest concentration of DON, whereas the innermost part of the wheat kernel has the least amount of contamination. There are some physical signs of mold damage. It is important to remember, however, that these signs are only indicators that mold damage has occurred. It is not an indication of toxin contamination or toxin concentration. This slide shows this fact in detail. On the right-hand portion of the slide, the wheat kernels at the top show healthy wheat kernels. The kernels at the bottom, however, show the pink-tipped kernels as well as some tombstone kernels. These two characteristics, the pink tips and the tombstone kernels, are physical signs of mold damage. The tombstone kernels occur when mold has entered the kernel and consumed the carbohydrate source in the middle. This results in a very lightweight, hollow kernel that has been named a tombstone kernel. The corn kernels on the left side of the slide represent some research on single kernel corn we performed at Trilogy. The very damaged kernel was analyzed and found to have 0.6 ppm or 600 parts per billion DON. Right next to it, the yellower, healthier looking kernel was analyzed individually and found to have 7 ppm or 7,000 parts per billion. So the healthier looking kernel had way more DON than the kernel that looked much more damaged. The physical characteristics of these two kernels do not correspond at all to the toxin concentrations we found. The important part of this slide is to understand that only testing can confirm the type and the amount of toxins that are present in a sample. While we are emphasizing that only testing can truly confirm toxin presence and concentration, there are some physical guidelines that can be evaluated to determine if mold damage was the potential that could lead to possible toxin contamination. Many purchasing points will put out some guidelines in place to limit acceptance of lots of wheat with increased DON potential. For instance, perhaps limiting scabby wheat, which is a term for the tombstone or pink kernels that you see in wheat, that those not exceed 2 to 3% of the total kernels evaluated. Also, low test weights per bushel are also an indicator that molds may have been present. Remember, those molds had to have a food source, and those come from the kernels. If the growing season where the wheat originated was cool and wet, this is yet another indicator that the toxins could be problematic. And remember, if the point of purchase brings in wheat mids, those are always suspected as having the highest toxin concentration. As many of you have either already implemented a testing program or perhaps are contemplating implementing a testing program, I want to take this time to highlight some of the testing challenges you may face, regardless of how you are performing the actual test method. First, let's look at the bar graph. This bar chart represents the total variability of a mycotoxin sample. The bottom blue portion of the graph represents the sampling portion of the process. Sampling includes how the sample is actually procured. There are many sampling diagrams available for everything from trucks, rail cars, and silos to feed bags, bunkers, or ship containers. The method that these vessels are sampled is important. As we have been talking, mycotoxin contamination is very inhomogeneous, so it takes some work to get a representative sample. How many probes will be taken? How much sample will be taken in each probe? What happens to the samples when they are compiled? How will these probes be compiled? All of these components of sampling are important. The red portion of the graph shows the variability that the actual preparation of the sample adds to the total result. This covers everything that is done to the sample after it's collected. That typically takes place in your laboratory. Things that affect this part of the process include how finely the sample is ground. Is every kernel sampled? How much analytical will be extracted? What extraction solvent is the one to use? How long will the sample be extracted and how? By hand shaking? By a blender? These are all critical parts of sample preparation that lead to the total variability of the answer. The green portion, which you can barely see, is the variability that actually comes from the test method. Some methods, such as LCMS-MS, may have an overall lower variability in the method than perhaps a rapid test. However, you can see that this portion of the process is not what makes up most of the variability. In summary, sampling, or how you get the sample, makes up about 61% of the total variability. Sample prep, or what you do in the lab to prepare the sample, makes up about 35% of the variability. And the actual test method, only about 3% of the variability. These numbers vary by specific toxin, what the actual toxin concentration is, and the commodity, but you get the general idea. To further show the sampling part of this variability, the table above the graph shows an actual study done with corn that contained approximately 20 parts per billion aflatoxin. The left column of the table indicates how much sample was probed from the truck. As you can see, the truck was probed numerous times. The right column shows the variability when multiple samples from that probe were analyzed. As you can see, the bigger the probe, the smaller the range of results. This shows that a larger probe when sampling vessels is important. The smallest sample probe resulted in a variability from less than 1 to 46.9 ppb. The results you get will be much better with a properly obtained sample. What we call the ppb challenge is the next thing we'll discuss. Mycotoxins are quantitated at the ppm, parts per million, and ppb, parts per billion level. Think about the small amount of material we're looking for. A ppb is 1 second in 32 years. There's some other examples, but that one is just really definitive of what a small amount we're quantitating. Maybe it seems like we're looking for a needle in a haystack, but when we recognize that these parameters affect our total result, it will help us get the best sample possible. Next, and we have hit on this challenge already, in the previous slides is the distribution challenge. Mycotoxins are not like protein, fat, or fiber, or moisture. A lot of those parameters we understand when we analyze commodities. Because mycotoxin contaminations happen where insects have damaged kernels or we find broken kernels, it's very inhomogeneous. Only 1 out of every 200 kernels of corn may be contaminated. If we're looking at cottonseed, only 18 seeds out of 150 were contaminated. You get the idea. You have to sample a lot of grain kernels. With this background knowledge, let's look at some sample prep videos to help explain this concept even further. Sample splitting is a very common practice, and it works well for the analysis of something like moisture. But let's look at what happens when whole grain is subsampled for mycotoxin testing. The picture below shows mycotoxin contaminated kernels in among healthy corn kernels. Let's see what happens when we riffle this. This is a standard riffler, much like you would see sample splitters. We're going to take one contaminated kernel and place it in 199. As the sample split takes place, there's no way that the sample on each side is going to have an equal amount of mycotoxin. This tray will clearly have more toxin than the total sample. The next tray will have no mycotoxin. So one of those samples will be a false negative and the other will be a high positive. Let's see what happens when we do 5 kernels in 925. We'll take those fluorescent kernels and mix them around before we split. These splitters are very similar to what you might find in a grain facility. Let's look at the results. In this container, we're going to see 1, 2, 3, 4 kernels representing kernels with mycotoxin contamination. This means that in the other split, there is only 1 kernel with mycotoxin contamination. This represents a low positive and a very high positive. Now let's look and see what happens when we finally grind the material. The material that we're grinding is the 5 kernels in 925. We're grinding them to a fine consistency and we're going to subsample. As we can see in this diagram, all of the fluorescent particles of that piece of corn or those pieces of corn are evenly distributed. Let's talk about that final result. What goes into the accuracy of that number that you are going to make your decisions on? Let's take an example of a wheat sample that was found to contain 3.2 parts per million DON. This level may be a result that is on the borderline of being a level that is either accepted or rejected at your point of purchase. Don't forget, this number represents a financial decision. If this number is too high for some reason, perhaps you are rejecting a product that you should accept. And on the other hand, if this number is too low, perhaps you are going to incorporate a wheat that has a higher level of DON into a finished product and be forced to either perhaps issue a recall or deal with some other out-of-spec product that you're going to have to make decisions on. This overview of result variability shows areas that contribute to what we call kind of the wiggle in our results. We have talked at length about some of those items, but as you go forward, remember to take into consideration things like technician training. Were they all trained well? And is everyone doing the same procedure every time? The testing frequency. How often should you test? What about the method suitability? A lot of times, a method that works well on wheat won't work well on, say, something like spices. And what about the particle size of your analytical sample? Remember, smaller grind size is going to give you more particles. Confirmation of sample results. It's always a good idea to take a certain number of results and confirm them to make sure that you can repeat that result. What about equipment calibration and performance? Do you have some ideas on how to routinely check those parameters? These are only a few of the things that make up an accurate result. Your system will have items that are not listed here. As we wind up the section on general things that will help establish a solid testing program, remember, a testing program will help mitigate some of the risk inherent in mycotoxin testing and provide you with the best data to make those important decisions. As a review, remember, with so many things about mycotoxins that are out of our control, things like the weather, our suppliers, the commodity you have to work with, it becomes even more important to control all of the things that are in our control. Things like how we do sample prep, how we train our staff, how we confirm our results, etc. Next, educate yourself and your staff about parameters that are out of your control. Can you watch weather patterns and determine if problems are a possibility? Also, look closely at your commodities and know what toxins are most likely to be problematic. Set some guidelines for acceptance criteria on physical characteristics, perhaps like bushel weight. Finally, focus on simple but important elements. Molds thrive in moisture conditions above 14%. Dry products before storage and ensure that no hot spots of moisture are being formed. Make sure transportation time, temperature, and moisture is not compounding the problem. Know the requirements of your clients and what toxin levels are critical to them. Don't forget, perhaps human consumption products may not have the same requirements as those for animal feed. Or maybe wheat midds that are used simply as a fiber source may be able to have a higher level of DON. All of these things are important components of a solid testing program. Reviewing these items ensure that you are setting up a system that has the final result in mind from the very beginning. Now, let's take some time to talk about the actual testing options. It's important to choose a rapid test method that accommodates your specific needs. You need to look at things like the physical space you have to do the testing. What type of a staff do you have that will perform the test? Evaluating these specifics will lead you to make the correct choice on which test fits your needs the best. There are a variety of different methods. Let's talk a little bit about two rapid tests. First, we'll talk about the lateral flow methods. These methods use a small plastic device that can be read as a single sample. These devices have many benefits. First of all, they are very rapid. Many times, it's only minutes from the extraction of the sample to the final quantitation. They are extremely easy to use. There can be on-site training or actually web training can be used because the methods are easily learned. There's no dedicated expensive equipment. Typically, what is used are pipetters and some type of a reader, and in many instances, a cell phone can be used for quantitation. These methods are nice because you can run a single sample. There's no need to use unnecessary sample wells if you're only running one sample. The calibration curve is already programmed into the reader or the phone. These tests are great for the point of purchase places when they receive many lots of materials at different times. The next type of rapid test also has a lot of benefits in a little bit different situation. ELISA tests, like the picture on the right of this slide, are great for running many samples together. Many times, depending on the well size, 20+ samples can be completed in one run. Another very nice thing about this technology is if you are in a facility that has this type of sample load, automation is available. Large numbers of samples can be run with little intervention because the robotics do the pipetting and the instrument will let you know when the results are available. Additionally, typically with these ELISA formats, data is downloaded to a computer, so more complex data manipulation is available for those facilities that need this type of testing. Regardless of what type of methodology you choose, testing for mycotoxins is here to stay. Currently, mycotoxin testing is performed by many companies in a lot of countries around the world. Many of those countries already have regulations in place, and other countries are implementing those new regulations for things that they import. The methodology around the world varies from a very rapid qualitative test to very sophisticated quantitative reference methods that are performed typically just in laboratories. There are also trends for lower and lower detection limits. And with global trade seeing more commodities going into new countries and new products being developed, mycotoxin testing is on the rise. In the next presentation, you will see a rapid mycotoxin test and the ease at which it is performed. Selecting a rapid test is a critical part of your total program. At Trilogy, we developed our quality system with analytical methods, but we realized that we also needed additional tools to round out our total quality system. We developed tools for our internal use, and soon we were being asked by our analytical clients to provide these tools to them so they may use them with their rapid on-site methods. Now, our quality tools include reference standards, known quality control materials for training and run acceptance, a laboratory shaker, purification columns, proficiency samples, just to name a few of the tools. We pride ourselves in helping other companies set up the best quality system for their mycotoxin testing. In our mind, helping others to obtain accurate mycotoxin results helps everyone in the end. If we can help in any way, please don't hesitate to contact us for more information on any of the topics we reviewed today. Thanks so much for your attention. We look forward to any of your feedback or questions.
The next segments will be dedicated to analytical procedures using instrumental techniques. And we know that mass spectrometry-based methods coupled with chromatography techniques, whether be it liquid chromatography or gas chromatography, constitute methods of choice for the determination of contaminants in food. In this next segment, we will have the privilege to have with us Dr. Gianni Dubois, who was one of the key instructors in the development of a capacity building program destined to a number of colleagues from the African continent. Dr. Dubois will review with us some of the principles, the requirements related to single contaminant detection or multi-residue, multi-compound determination using liquid chromatography coupled with mass spectrometry-based techniques.
In Section 1, we look at single residue methods and the factors taken into account during method development. Traditional analytical methods were developed for the determination of a single or a small number of contaminants at once. With the advent of LC-MS, methods that determine the presence and quantity of large numbers of analytes are commonplace. While the multi-residue methods provide speed and cost savings, some analytes are not compatible enough with large groups of other analytes to be included in a multi-residue method. In addition, some analytes require special sample preparation steps that may not be optimal for multi-residue methods. A method needs to be selected with a thought process of the sort shown here from the Codex Procedural Manual. It must be able to measure the contaminants of interest, in this case lead. Then, we ask if it's applicable for the matrix, in this case juice. Then, whether it can achieve the LOQ of interest and generally meets the performance requirements for our purpose. There are many performance requirements to evaluate. We will review this topic in Module 9, but briefly, we have to look at trueness, applicability, limit of detection, limit of quantitation, precision, repeatability, intralaboratory, and interlaboratory reproducibility, recovery, selectivity, sensitivity, and linearity. A method typically comprises steps for sample preparation, including homogenization and extraction, purification, adjustment of the concentration either through concentration or dilution, and measurement. A single residue method is one that measures only one component, but sometimes a group is considered one, like aflatoxins, for example. In the case of aflatoxins, there are 4 separate aflatoxins, and we could measure them together as one, or individually, depending on the measurement technique that we choose and the purpose. Some definitions are important at this stage. In general, indication is the result of a screening method. Identification is a qualitative result obtained from a highly selective method, and confirmation is the agreement of results from 2 or more independent analyses. Screening methods are very important in food safety because of the large number of samples and an increasingly larger number of contaminants covered by regulations. Screening methods are typically rapid, inexpensive, and reliable, and they can be single or multi-residue methods. The main disadvantage is that the result must be confirmed. In most regulatory systems, all positive screening results must be confirmed through a second analysis before regulatory action can be taken. This is to avoid acting on a false positive result. The number of negative results that are confirmed is more variable. It's important that enough negative results are confirmed to ensure the reliability of the screening method, in this case, a low level of false negatives. This topic will be revisited in Module 9 on quality assurance systems. A method used for the identification of a compound can be indicative or must confirm this identity. It's important to know what the terminology means in your regulations. We use a set of criteria that have been assigned point values to determine if a method is just for indication or if it is for confirmation. An identification in the United States must score at least 4 points, while confirmation methods need to score at least 5 points. First and foremost, the points are based on a comparison with a reference standard used in the same run as our samples. We cannot use a library or historical data for comparison with a current chromatogram. Codex issues guidelines on the use of mass spectrometry for the identification, confirmation, and quantitative determination of residues, which is identified by this document number. The reference at the bottom of the page is to the library of guidelines issued by Codex, including this one. These are criteria associated with the chromatography. In other words, 1 point is allocated if we have a peak at the same retention time as our reference standard. If we have a single detector and run the sample on 2 different columns, we can accumulate 2 points. When using a mass spectrometer as the detector, then there is a point for the precursor ion, or when using MSMS, there is a 1.5 value for each transition. As we mentioned before, a transition is the combination of a precursor and a product ion. With high-resolution mass spectrometry, there are 2 points per precursor ion and 2.5 points per transition. Consequently, a matching retention time and 2 transitions provide enough certainty for confirmation in LC-MS. A confirmation is a demonstration of results in agreement with those obtained using an independent analysis. In the points system we just saw, the chromatography and mass spectrometry are considered 2 independent analyses since they use completely different principles. 2 columns are also considered 2 analyses. In the point system, we want 5 points for confirmation. Codex has published a list of criteria for confirmation using different methods of analysis. The rationale is the same as in the example from the previous 2 slides, but it is less prescriptive as Codex lets each authority decide the specifics of how many points they want. As a general rule, only the combination of chromatography and mass spectrometry provides enough evidence in a single analysis to be considered a confirmed result. LC diode array can be enough if the whole spectrum is characteristic. The others all need a second analysis to confirm. In the criteria associated with the chromatogram, the peaks not only need to appear at the same retention time, but also need to have similar intensities for similar concentrations of standards. These 2 tables compare criteria from the European methods and from the US FDA. We will review these in more details in the in-person section of this course. There are also quantitative criteria associated with the ion ratios. If the ion ratio is not similar under the same ionization conditions, there is a good likelihood that the compounds are not the same, even if they share a precursor and a product ion. Let's go back more specifically to the method development steps. We need to select diagnostic ions for our compound of interest. We know that the collision chamber will create many different product ions, so we need to choose wisely. A good ion is one that is specific to the fragmentation of the compound of interest. In other words, it contains important portions of the original molecule. It should not be common from the fragmentation of a lot of molecules. In the example illustrated here, we have the precursor ion in the center and a number of fragments with relevant chemistries. The mass spectrum shows the peak intensities for all of them. We would want to use ions with a high intensity because the intensity affects our limit of quantitation. So we might choose the transition 305 to 169. There are minimum values of peak intensities to look for. This is in the chromatogram. A good primary ion has a signal-to-noise ratio of at least 10 to 1. This is the minimum for quantitation. The second ion should have a signal-to-noise ratio of at least 3 to 1. If there is an isotopic cluster, we should not use more than 2 diagnostic ions from the cluster. More than that is just redundant information. Finally, in LCMS, we have only one molecular ion, and we should always use it as our precursor. As we discussed in Module 5, we can have 2 in GC, so we could choose which one to use for MSMS or both for the GCMS method. Let's look at an example here. This is perchlorate in melons. The ion ratio of the secondary over the primary diagnostic ions is just under 34% in the standard. And in the sample, it varies from 29 to 37%. The 2 columns on the right indicate that these ions would be acceptable both in the US and in the EU. Let's review a few examples of how we calculate the score, or the number of identification points for methods, to decide if we have enough for identification. In this first example, a result is submitted to us which contains 3 ions from low-resolution GCMS in the sim mode and a retention time match of the standard and sample. Is it an identification? Yes, it scores 4 points. Here, it's a single MS, so precursor ions are all 1 point. 1 identification point for each ion, and 1 identification point for the retention time match, for a total of 4. In this second example, we have a retention time match of sample and standard on GCFPD and GCXSD using the same column, and a retention time match on an alternative GCFPD and agreement of quantitation within plus or minus 30%. Do we have identification? The answer is yes. This has scored 4 points. 1 identification point is given for each alternative detector, plus 1 identification point for matching retention time on alternative chromatography systems, plus 1 identification point for the agreement of quantitation by 2 independent methods, for a total of 4. These 2 examples showed that MS is not the only instrumentation that can be used, especially since it can be difficult to deploy in laboratories where the electrical supply is not stable. If we use 2 non-mass spectrometry methods, then residue values between the 2 independent methods should not be significantly different. In an ideal world, we would be able to verify our method with mass spectrometry for confirmation and see that we obtain the same results. If possible, we also need to verify these parameters with incurred samples. An incurred sample is one that is naturally contaminated as opposed to a laboratory spike. Recovery determination using fortified samples is not sufficient. Finally, the reference standards should be certified if possible, or at least verified by an independent analysis. Let's look at a real-life example with the data now. This work was presented by Dr. Alex Kunitsky and Zhong Wang of the US FDA. We are looking for diazinon in ginseng. Diazinon is an organophosphate insecticide. As a side note, ginseng is a difficult sample. That's why it's a good example for limitations of methods. In GC with an FPD detector, we have a peak at 23.75 minutes, and our reference standard has a peak at 19.6 minutes. In GCMS, we have a peak in the chromatogram on the top right at 20.15 minutes, and we see the overlapping peaks of our internal standard and ions for diazinon. So, what can we conclude? The GC with 2 single ion monitoring by MS is enough for identification. When combining the results of GCFPD and GCMS with 2 single ions, and observing the matching retention time, then we have a confirmation. In this second example, we are looking at GCFPD of a carrot extract containing low concentration of chlorpyrifos, another organophosphate pesticide. The GCFPD chromatogram shows a retention time of just over 28 minutes. Now we have the mass spectrum on the right and the 3 chromatograms from the single reaction ion monitoring for mass to charge ratios of 314, 258, 260, 286. The problem we see here is an interference with 2 of the ions. So we go to GCMSMS, and for the same sample, we were looking at the transitions of 314 to 286, and 314 to 258, and 314 to 250, and 197 to 169. All of them have very high signal-to-noise ratio and no interference. Now we look at the GC time-of-flight mass spectrometry chromatograms. Low microgram per kilogram chlorpyrifos in carrots is observed again. I should mention that all of these chromatograms in the previous slides and this slide were acquired from incurred samples. So the findings in this case are that GCMS single ion monitoring alone did not satisfy identification criteria, since only 2 diagnostic ions with the proper ion ratios were free from interferences. GCMS single ion monitoring combined with FPD did satisfy identification and confirmation, since they are 2 different detectors and the retention times matched for chlorpyrifos. GCMSMS and GCTOF-MS each alone satisfy the identification criteria, since diagnostic ions with the proper ion ratios were free from interferences. These also satisfy confirmation criteria. Last but not least, the LOQ and LOD of a method need to be calculated. We will do this in our laboratory session, but here is a good reference. Briefly, we calculate the limit of quantitation, LOQ, as the concentration that provides a peak with a signal-to-noise ratio of 10 to 1, and LOD at a signal-to-noise ratio of 3 to 1, and then we back calculate how much was in the original sample before it went through sample preparation, which may include concentration of the residue. The point of showing the results here is to compare the values of LOD for different techniques. If an MRL is really low, there may not be very many techniques to measure it. Advantages and challenges of multi-residue methods. Let's start with a summary of why MS has become so popular. First and foremost, it enables us to do multi-residue analyses, which means that we get sometimes 200 or even 300 answers with one test. Sample preparation can be easy, especially with the catcher's techniques. It can be relatively easy or cheap to deal with matrix effects. I say easy or cheap, usually not both. Easy is to use isotopically labeled standards, and cheap is more work with matrix-matched calibration. But then you have to make a calibration for every matrix, or test if you can mix matrices together to make a more general matrix-matched calibration. We will talk about this further in the hands-on portion of this training. Finally, the fact that it accumulates enough points to obtain a confirmation of identity is also quite practical. Historically, there were completely different extraction techniques and combinations of detectors and columns on GC. Multi-residue methods pose a number of challenges. For example, the sample preparation method must extract all compounds of interest. The cleanup steps must remove problematic co-extractives, but do so without losing any of the compounds of interest. Using a tandem quadrupole instrument reduces the level of cleanup needed in a wide variety of samples, but there is still a need to remove any analytes that could negatively impact the ionization in the source, especially if it increases the limit of quantitation and the limit of detection to above the levels needed for the purpose of the method. The sample preparation with suitably broad retention must lead to an extraction efficiency meeting regulatory requirement, as discussed in Lesson 2. It must not cause any degradation of any of the compounds of interest. When contaminants are present in vastly different concentrations, it may be necessary to include steps of concentration or dilution in order for the signal to fall within the linear range of the detector. Finally, it has become expected that multi-residue methods will involve relatively easy and fast sample preparation. It's overall a tall order. Similarly to the GC sample preparation tree, there are many different versions of sometimes relatively similar sample preparation procedures for liquid chromatography. The graphical representations of the genealogy of sample preparation methods for pesticides was prepared by colleagues at the US FDA, Drs. John Wong, Alex Grunitsky, and Kai Zhang. Let's look at an example of how sample preparation procedures have changed in time for MRMs. Sample preparation procedures used in the 1970s for the determination of halogenated pesticide residues required 400 milliliters of hexane and using a sock slit for extraction for 8 hours. The extract was then concentrated down to dryness and reconstituted in 20 mL of hexane. The next step was to run the sample through a fluorosil column to separate fats using 200 mL of solvent and a silica gel column to separate pesticides from PCBs using another 500 mL of solvent. The quantitation was performed using gas chromatography with an ECD detector, and confirmation was done by GCMS. This method used over 1 liter of organic solvent, in this case hexane, per sample for the measurement of a few halogenated pesticides. In comparison, the catcher's methods, described in the figure on the right, uses 15 milliliters of acetonitrile for LC and an additional 1.5 milliliter of toluene for GC per sample for the measurement of 20 pesticides. Modern variations on the catcher's method use the same volume of solvent for the measurement of upwards of 200 and even 300 pesticides. Besides being easier to perform, faster, and less costly, these methods are better for the environment. The LC-MSMS multi-residue method needs to be set up in order to separate the analytes sufficiently to enable MS analysis and produce peaks that are a compromise between sensitivity, meaning narrow, and wide enough to allow enough time for the mass spectrometer to go through all the components of interest that elute in the same retention window. The effect of the selection of mobile phase on all analytes of the method must be understood. We discussed examples of the effect of using a gradient and different mixtures for isocratic runs in Module 4. The effect of pH is also important to produce symmetrical peaks and favor ionization. The column also has to accommodate all analytes. Luckily, food contaminants analysis has grown globally and now represents an interesting market for column manufacturers who now market columns with optimal performance for these applications. This example illustrates the effect of the mobile phase on the measurement of mycotoxins. Chromatograms in A were acquired using methanol, while acetonitrile was used for B. The separation is cleaner in B. From the same article, we can look at the figure showing the effect of the column on the separation. There is rarely a perfect answer, especially because we are constantly adding analytes to our methods. In this case, the three original mycotoxins of interest, namely DON, fumonisin, and nivelenol, are perfectly separated in B, but some of the other peaks are not. None of these three columns used in the conditions shown produce a perfect separation. Luckily, the mass spectrometer can resolve two or more overlapping peaks. This publication is available for free from its authors on ResearchGate. The real new topic in this section is the concept of running more than one analyte at the same time in the mass spectrometer. It is not strictly what happens, since only one mass-to-charge ratio is allowed through MS1 in the MRM mode, but it describes the need for sharing of a time frame by two or more analytes. This slide shows a typical software window for a multi-reaction monitoring experiment in MSMS. The green boxes span the portion of the time axis located at the top. Data is to be acquired for each of the pesticides listed on the left and including two product ions identified here as mass pairs during this time window. In this experiment, the period between, for example, 5.5 and 6.5 minutes shows 8 analytes. Consequently, MS1 will need to allow 8 different precursor ions through. The collision chamber will need to be energized at the best collision energy for each of them to produce 2 product ions. And finally, MS2 will need to filter 16 product ions onto the detector. This particular method includes over 150 pesticides, so there may be more than 16 overall. Some of the MS parameters are fixed during a run, such as the ionization flow rate at injection, desolvation gas, and source temperature. Other parameters can be tuned for each analyte, such as ionization current, positive or negative, and the collision energy for fragmentation in the collision cell. Now comes the concept of dwell time. Dwell time is the amount of time that the mass spectrometer spends accumulating data. We usually report dwell time and resulting points per peak, where a minimum of 10 to 15 points per peak is preferred. During the period of accumulation of the information, the dwell time, the signal is average, but so is the noise. This results in an increased signal-to-noise ratio since the noise is random and will add and subtract, while the signal should always be about the same. Of course, a greater signal-to-noise translates in lower limits of detection and limits of quantitation. In summary, we are working with very low concentrations in food safety, so we need to compromise in a way that provides a good enough signal-to-noise ratio to meet the LOQs required by the regulation. Circling back to the dwell time, this is a value that can vary between instruments, but generally not by an order of magnitude. Using round numbers for illustrative purposes, an analyte may produce a signal-to-noise ratio of 3:1 with 10 millisecond dwell time. This would be the absolute minimum dwell time to use to make a screening measurement, which needs a signal-to-noise of 3 or more. In theory, the signal-to-noise ratio improves according to the square root of the additional time. So moving to 100 millisecond dwell time should improve the signal-to-noise ratio by 3.16 times. Considering the margin of error, this is approximately the dwell time that would be needed to obtain a peak with a signal-to-noise ratio of 10, the minimum requirement for a quantitative measurement. Putting all these considerations together clearly emphasizes why there is a limit to the number of analytes that can be included in a multi-residue method. It could be possible to increase the number of analytes included in the method by lengthening the experiment in a manner that would cause better separation of the analytes. A sufficiently long dwell time could then be allocated for each analyte in a very large method. At this time, methods including upwards of 300 pesticides are some of the largest methods for regulatory testing. As mentioned before, regulatory testing requires a high degree of certainty because it will trigger regulatory action that is very costly. Screening methods comprising of around 1,000 pesticides are advertised, but they are generally not very popular because the preparation of the calibration curve for such a large method is both arduous and expensive. In conclusion, there is no such thing as a universal MRM. Analyte characteristics such as whether they are volatile or not, polar or not, or sensitive to degradation at high temperature fundamentally make the universal method impossible. More practically, there is generally not a need for exceedingly large methods because many laboratories have a restricted mandate that can limit the scope of commodities analyzed, the list of pesticides of interest, or both. When the concern is with completely unexpected analytes, high-resolution mass spectrometry has a better fit for the purpose. I wish to acknowledge contributions from many people to the material presented in this section.
We hope this broadcast offered you a good perspectives on various facets of food contaminant detection. We certainly could not cover all the areas of priorities and interest in this particular field of food regulatory science. Definitely, we welcome your input and your comments as to future areas of interest that you would like us to cover as part of the GFoRSS TV broadcasts. Once again, I would like to thank all those who contributed in the development of the programming. I would like also to thank particularly Dr. Eli Bouyazbek, who has directed the development and the availability of this material as part of GFoRSS TV, and also, most of all, you, those who are with us live today, and those who will be following this broadcast subsequently. Thank you very much, and we wish you a great continuation of the day.
The Global Food Regulatory Science Society. GFoRSS mission includes knowledge generation and dissemination, underpinning food regulatory decisions, in support of food control systems, Our interventions focus on promoting food regulatory scientific disciplines. Consistent reliance on food regulatory sciences enables robust food control programs, contributing to more resilient food systems and a safe, accessible, abundant and nutritious food supply.